So now we're gonna segue a bit. It's my delight to welcome Dr. Steve McCarroll, who's the Dorothy and Milton Flyer Professor of Biomedical Science and Genetics. He's the Director of Genetics for the Broad Institute, the Stanley Center for Psychiatric Research. And his lab really focuses on how genes and cells make the human brain work and why the human brain is vulnerable to specific disorders such as schizophrenia, bipolar disorder, and neurodegenerative disease. It's rare that you get to say this, but I get to say this. His lab invented and freely shared now widely used technology called DropSeq for analyzing gene expression. He's won a number of awards, including the National Alliance for Mental Illness Scientific Research Award, the Smith Family Foundation Award for Excellence in Biomedical Research, and he's had a number of very large grants from NIH. And of course, he's been very productive and really has done some outstanding work. And while we welcome him to the stage, I'm going to load his talk. Welcome, Dr. McCarroll.
Great. All right. Well, thank you for that kind introduction and for inviting me to come and share this work. Awesome. Thanks. Oh, and disclosure. So I and scientists in my lab are inventors on patents and patent applications related to work that I'll talk about in this talk. So biology has more than its share of fictional monoliths, but probably none is more pervasive than biology itself. Although biologists set out to find universal principles that underlie the function of the brain and other organs in all living things, in fact, it's very clear that there's a lot of variation in biology from species to species, at a very different scale between males and females of the same species, and fundamentally just among every living person, which is one of the reasons why human genetics is starting to find bona fide genetic influences on risk of these disorders. And two particular aspects and so biological variation is also a potentially powerful tool for beginning to understand the biological basis of various things. And so today I want to talk about efforts to understand two of these things. One is sex differences in risk of illness among humans, which I'm going to talk about trying to understand through the prism of human genetics. And the second is about species differences in brain circuits and behavior, which I'm going to talk about through the prism of single cell analysis. So men and women actually have some very different collections of vulnerabilities to different disorders that we really don't understand the biological basis of at all. And I'm going to start with two examples. One is lupus and a couple of other autoimmune diseases. Females are profoundly more vulnerable to lupus. So the ratio of incidence between females and males among adult onset cases is something like 9-to-1. And that's not true among child onset cases or among onset later in life. It's really something about adulthood and the reproductive years that women are profoundly more vulnerable, but we don't know why. In schizophrenia, it's quite the opposite. Males seem to be more vulnerable, to have more severe negative symptoms, less remission, more relapse. And while estimates vary about relative incidence of males and females, it seems to be the case that among those cases that onset early in adulthood, that estimates kind of approach 2-to-1 in terms of male to female vulnerability. So there's a lot of sex-specific risk for complex polygenic illnesses, and we don't know why this is the case. So it was once hoped or even expected that it would turn out to be the case that there would be large effect alleles in chromosomes. And there are clear examples of this in monogenic brain disorders, such as mutations in MECP2 that are the cause of Rett syndrome. But for complex polygenic disorders that are shaped by many different genes and by environment, the sex chromosomes actually have a small fraction of the alleles that are implicated. And so this doesn't just boil down to sex chromosomes. Instead, there seem to be aspects of male or female biology that are protective against these illnesses. And there are many, many theories about what those might be. But it's been very hard to determine what is actually important. So a few years ago, we described the genetic basis of the largest common genetic effect on risk of schizophrenia to emerge from human genetic studies. So if people have followed the human genetics in this area, schizophrenia is a highly polygenic disorder. As human genetic studies have expanded, they've mapped genetic influences on every human chromosome. But is that? Oh, all right. For a moment, I thought I was a subject in one of Yale's experiments. There are influences on every chromosome, but the strongest influence by far has been in the major histocompatibility complex locus on chromosome 6. And because that's the part of the genome that has so many immune system genes, it was assumed for many years that this was potentially trying to tell us that maybe schizophrenia, maybe it's an autoimmune illness, maybe there's an infectious element. And what we found, I'm not going to sort of go over the whole story, but what we found is that actually this genetic signal arises in substantial part not from the HLA genes that were kind of assumed to be the basis of this, but from a different immune system gene that's also just happens to be present in the same part of the genome as those genes, which is the complement component 4 genes. And what we found is there's many different forms of the complement component 4 locus. So these 2 different complement component 4 genes, C4a and C4b, and different people inherit different numbers of different numbers of C4a genes and different numbers of C4b genes. The genetic variation also causes them to be expressed at different levels. But the common theme we found is that alleles that cause you to have more expression of C4a increase your risk of schizophrenia in rough proportion to how much expression of C4a they drive or generate. It's still just a quantitative change in risk. This isn't like big B, little b genetics or Huntington's genetics. It's still just one of many genetic risk factors. But one of the things that distinguishes this from all these other loci is that here at least we now appreciate what the alleles are and what the first order molecular effect of the alleles is. And that kind of tapped in an interesting way to some things so the complement system has long been known for its role in the innate immune system, it actually tags debris, cellular debris, for phagocytosis and destruction by macrophages and other immune cells. And our colleagues, Beth Stevens and Mike Carroll, really interesting work from their labs has shown that C4 these same molecules are also used in the brain to contribute to the pruning of synapses during brain development. And that sort of invites this idea that given that what we found is that it's sort of overactive or overproducing alleles that generate the greatest risk for schizophrenia, that potentially is mediated by exaggerated pruning or excessive pruning or pruning that's hard to turn down at appropriate times. So I mentioned lupus a couple of slides ago as an illness that has a very opposite distortion in sex incidence. So lupus is an autoimmune disorder with recurring symptomatic flares. It affects many different organ systems. The common theme is that patients develop, and this is what's diagnostic, patients have developed autoantibodies against ribonuclear proteins. So these are proteins that are on the insides of cells that the adaptive immune system wouldn't normally see, but somehow they've developed autoantibodies against them. The cause of lupus is unknown, and there's no cure. So it's just treated by trying to suppress the immune system with steroids and other immune suppressants. So as with schizophrenia, the strongest common genetic association in lupus is also in the MHC locus. It's quite a bit stronger than the other ones. In fact, in this paper, they used a broken axis just to kind of fit it all on the same plot. But it's the strongest genetic influence in lupus as well as schizophrenia. And in large part, as you would expect for an autoimmune disease, it's been assumed for 20 years that this must arise from the HLA alleles which present the various antigens to the adaptive immune system. And that's been the kind of prevailing belief for the last couple of decades. But there's always been something about this that never quite made sense, which is that unlike diseases like type 1 diabetes, for example, where there are specific HLA alleles that present the disease-critical autoantigen, there's never been any clear association in lupus of specific HLA alleles with specific autoantibodies. And so we actually wondered if, as at schizophrenia, this might actually not be due to HLA alleles at all, but might potentially be due to these complement component 4 genes. And so we developed a way to analyze this C4 genes from emerging whole genome sequence data, which is now being produced in abundance by many different large-scale sequencing projects so that we could tell what form of the C4 gene each person had. And then we developed ways to relate each of those C4 alleles to surrounding SNP haplotypes, because each of these alleles, when it arose on a specific chromosome with a specific set of other genetic variants around it. And what this lets us do is just kind of turn a molecular biology problem into a big data problem, because there's these vast human genetic studies in which thousands of people have been genotyped using SNPs, so their SNP data is available, and this just lets us infer which C4 alleles people have from their SNP data. So when we applied this analysis to lupus, what did we find? So applying this analysis to large lupus genetic studies, more than 6,000 cases and 11,000 controls. So what we found is the following. And this is kind of an unconventional way of representing genetic data, so I'm just going to walk you through it. So different people have different numbers of C4a genes, and different people have different numbers of C4b genes. And the 11 most common combinations are shown on this plot, and the size of the circle is the area of the circle is proportional to how many people have that particular combination in their genomes. But you can see there's florid variation. If you picked any two people, they actually probably have a different number of C4a and C4b genes. And then what we've done here is we've colored each of the circles by what we estimate is the lupus risk of people in that group. And what you can see is that these different groups differ profoundly in their risk of lupus. This is actually not a small effect at all. There's 8 or 7-fold variation among these different common groups in risk of lupus. But there's also florid variation, but with a theme. So you'll notice, or with a pattern. So you'll notice that if you walk from left to right across the plot, that is to say, if you control for C4b copy number but increase C4a copy number, that people's risk goes down profoundly. So we think that actually having more C4a is profoundly protective against lupus. If you walk along the plot along the other axis, so if you control C4a copy number and increase C4b copy number, you find that C4b is mildly protective against lupus. It's also protective, but it's just a smaller effect. But for both, it's protective to have more copies. And if you walk diagonally along the plot, that is to say, if you go to groups that have like one more C4a copy but one fewer C4b copy, you can see that that's also protective, again, suggesting that C4a is more protective than C4b is. So how would complement protect from lupus? So all the time in our body, cells die or are damaged, but particularly like if you get a sunburn or if you get an injury or things like that. This is something that's just happening all the time. And whenever that happens, cell damage exposes the adaptive immune system to nuclear debris in the context of a lot of cell injury and distress signals. And this is where the immune system goes looking for little critters, much like the microbes in Yale's talk. And there's the potential for it to get confused and see something it doesn't usually see and think that that's the problem. So these complement, and this leads to the potential to break tolerance, leading to autoimmunity against self-ribonucleoproteins. So what the complement system is really good at, in fact, what it was known for long before its roles in synaptic pruning, is that it quickly binds to ribonucleoproteins and facilitates their clearance, which attenuates the exposure of these proteins to the adaptive immune system. And that is why we think having the system be more vigorous is protective against autoimmunity, because the more quickly you can clear these things out, the more you can kind of preempt the exposure of the adaptive immune system to them. So, and I just want to mention very quickly, independent work by our colleague Mike Carroll and Leo Simone in his lab, and Mike's an immunologist, and they've generated mice with humanized C4A or C4B, and actually found that mice that are lacking C4 have defects in the clearance of apoptotic debris, that human C4A rescues that very strongly, C4B rescues it less strongly, and similarly that in a mouse lupus model, that the mice tend to develop autoreactive germinal centers with autoreactive B cells, and that happens to a much greater extent in mice that lack C4, but you can rescue that a little bit by giving them human C4B, but what really helps is to give them human C4A. And so this actually is the first biological evidence supporting what was a really surprising thing to come out of the human genetics in schizophrenia, which was that C4A and C4B are actually maybe very different in their biological effects. So, but now I want to come back to schizophrenia and the question that I started with, which is about sex-specific vulnerabilities. Because so what we've found now through the prism of human genetics is that C4A increases risk of schizophrenia and that it reduces risk of lupus. And as I mentioned at the beginning, schizophrenia tends to affect males more severely and more frequently. Lupus tends to affect females far more frequently. And so we just wondered, does this genetic effect actually differ between males and females, which almost no genetic effects are. So there's a long history of looking at this. Almost all genetic effects on risk of brain illnesses act with equal strength in both males and females. But what we found is this is a big exception. So each of these different C4 alleles has larger effects in males than it has in females, and that's true in both lupus and schizophrenia. So that is to say, this genetic variation matters in both sexes, but it somehow shapes risk more strongly in males than it does in females. So why could this be? Well, one possibility is if these proteins are differentially abundant in males and females, that is to say, if the system is just kind of being run at different levels, that could be the substrate on which genetic variation is acting. So in the brain, complement proteins are secreted by cells and rinsed into the cerebral spinal fluid, and CSF, on some level, gives a way of monitoring them. And in this study, in collaboration with Roel Afaf at UCLA, we analyzed CSF samples that had been contributed by 640 young, healthy research volunteers at the time they were undergoing surgery for like back surgery and things like that. So these are young, healthy research volunteers, and we measured C4 concentration using ELISA, concentrations of both C4 protein and C3, which is the immediate downstream effector of C4. And what we found is that males tend to have higher levels of both C4 and C3 in their CSF, and in particular, males are greatly enriched in the tails of the distribution of particularly people with particularly high levels. And this has also been noted independently in plasma that's sampled from adults, so that during adulthood, but not during the kind of reproductive years, but not before or after that males have elevated levels of complement proteins relative to females. And these age ranges align with the age ranges in which there's also sex-biased incidence of both disorders. So just to summarize what I've told you about so far, so C4A protects, we've found that C4A protects strongly against lupus, C4B protects more modestly, and I think this pleiotropy is going to be important to consider. There's a lot of excitement these days in considering the complement system for therapeutics for various disorders, but I think one has to proceed very mindfully of the fact that for some disorders, higher levels of these complement proteins actually may turn out to be quite protective, and so one would want to be very careful about any kind of therapeutic whose goal was to modulate their levels. Secondly, that males have elevated levels of C4 and its effector C3 in both the blood plasma and CSF, and that C4 alleles have stronger effects on male than female risk in both schizophrenia and lupus. And what we think this is pointing to is that there's just, for whatever reason, a more active or more vigorous classical complement system in males during the reproductive years. And this may protect men from lupus while increasing their vulnerability to schizophrenia. And finally, I just want to say about this set of results on C4. So there has a potential biological mechanism for these effects of C4 on schizophrenia involves this. It's this effect on synaptic pruning. And one of the reasons I think there's naturally interest in that is that it would offer a plausible connection among a few things. One is this genetic finding at C4. One is these observations by David Lewis and others that many schizophrenia patients have fewer dendritic spines on pyramidal neurons in upper layers of the cerebral cortex. These observations by Tyrone Cannon and others of excessive cortical thinning in patients, and also the fact that a lot of these processes of synaptic pruning and cortical maturation are taking place during adolescence and young adulthood, the kind of second and third decades of life, which tends to be the time of most frequent clinical onset of these disorders. And it may be that C4 and that synaptic pruning offers a plausible connection among all of these things. I think it's reasonable to be excited about testing that hypothesis. What I want to emphasize, though, is it's still just a hypothesis, and I think that's just very important to keep in mind. And just two other things I want to just make sure people are thinking about is that complement may also affect brain function by additional mechanisms that we just don't know about yet. Like the pruning thing was just found a few years ago. There may well be, these are very general purpose molecular tags. They're likely to do more than one thing. And secondly, that complement could be shaping, and even pruning could be shaping disease progression or severity rather than being the founding pathology, and we just don't know enough yet to know which of those is the case. So I just mainly want to say that we can all as scientists sometimes make this mistake of sort of we connect the dots that we see at any moment in time, but often more work finds new dots and a different, more plausible path for connecting them. And so I just want to emphasize sort of healthy agnosticism and sort of drawing the line between what we really do know and what's plausible and would make a lot of sense, but we really don't know yet. And then I want to acknowledge the many people who contributed to this work, but especially Nolan Kamataki, a really wonderful and brilliant graduate student who did all the human genetics work that I told you about, and also our collaborators in lupus genetics, especially Tim Weiss and Lindsay Criswell. So now I want to switch gears to present some recent things that we found on this other question about differences in different species, and through the prism of single cell analysis. But to kind of pivot, I actually want to just go back briefly to the Middle Ages, which is a time in human history that is probably fair to say is charming only in retrospect. But one of the charming things about the Middle Ages is that there was a synthetic theory of all disease, and that disease was called, or that theory was called the four humors theory, and the idea was that the body is a bag of fluids, four different fluids: blood, black bile, yellow bile, and phlegm, and these fluids are supposed to be present in certain molar proportions, and illness arises when they drift out of their natural proportions. That is to say, when we have a chemical imbalance. So if you had too much blood, you were sanguine. If you had too much phlegm, you were phlegmatic. Along came the 20th century, and this theory was replaced by a much more scientific-sounding theory. That theory held that the brain is a bag of neurotransmitters. Those neurotransmitters are supposed to exist in certain proportions. If they drift out of their ideal proportions, that's the cause of illness. And it should be clear that theories like this are sort of hopefully places along the way to better and better theories. It should also be clear that the brain is not just a bag of neurotransmitters. The brain tissue contains hundreds of specialized cell types. They differ dramatically in size and shape and physiology and biological mission. They use the same genome in profoundly different ways. And it should be our aim to understand function and genetic and dysfunction and genetic perturbation at this level. The challenge was that until recently, if you wanted in an unbiased way to measure all of the analytes in a tissue, for example, the level of expression of all of the RNAs, you had to first take this wonderful mixture of all these different cell populations doing different things and essentially throw it into a blender and measure the level of all the molecules in the smoothie. But that's a challenge because all of the interesting stuff is in the specializations, right? You can imagine if you were trying to study the way a city like New York works, and you could only do so by collecting some statistics about the average resident of New York. You would learn nothing because all the information is in the diversity and the specializations. So several years ago, we set out to develop a way to analyze brain tissue much more like a fruit salad, where you could probe and analyze and learn from each individual piece of fruit. And this is work that was led by Evan McCosco, a postdoc in the lab, who's also a practicing psychiatrist. Actually, I should say now he has his own lab, but Evan did this work while pursuing a research residency in psychiatry at MGH and McLean Hospital. So how could we analyze how thousands of individual cells are each transcribing their genomes to produce the RNAs that they use to do their jobs? So we developed a way to do this by by first separating the cells into vast numbers of tiny droplets. So the idea is to create an oil aqueous emulsion in which there's a very large number of aqueous droplets suspended in oil. And the nice thing about droplets is they're tiny. They're larger than cells, but they're tiny. You can fit millions of them in a centrifuge tube. And together with Oni Basu and David Waits and other colleagues, we designed and built microfluidic devices that could encapsulate large numbers of individual cells. Let me just see if I can play this movie for you. Each cell into its own tiny water droplet. And this is a zoom in on these devices. Each channel is about the width of a human hair, and the cell, you can see, will kind of quickly flow through here. And this is actually a barcoded bead that I'll tell you about in a second. And the idea is each cell lands in a droplet with its own little barcoded bead. The barcode lets us, the DNA barcode allows us to identify, to kind of tag each cell's RNA so that when we sequence all of them later, we can tell which RNA molecule came from which cell. And this movie is greatly slowed down. This is the actual speed with which cells can be encapsulated in the droplets. And it's so fast because the volumes are actually so tiny. So we needed a way to remember which RNA molecule came from which cell and which droplet. So we developed a way to address each droplet by delivering a different primers with a different DNA barcode to each individual droplet, and came up with a way of synthesizing millions of barcoded beads, each with a different barcode, by what we call a split pool synthesis, where we would take a set of beads, divide them into 4 groups, add a different DNA base to each of the 4 groups, then recombine them, then split them again, add a different DNA base to each of the 4 groups, and then combinatorially, if we just did that 12 times, we'd create millions of different beads, each of which was capable of delivering a different DNA barcode sequence to a different cell. So, and our colleague Rahul Satija in Aviv Regav's lab developed machine learning strategies for analyzing the kind of data that comes out of these experiments, and we found that cells could cluster into recognizable types and states based on these RNA expression patterns. So for example, in this analysis, which is of cells sampled from the retina, even if you hadn't known about all the different photoreceptors and interneurons and different cell populations that are in the retina, that the data would have just classified the cells into different types that end up corresponding in a clear way to rod photoreceptors and cone photoreceptors and things like that. So we made DropSeq free and open source. We wrote detailed 30-page protocols and placed them on our lab website. They've been downloaded tens of thousands of times. There's online discussion groups where people help each other, analyze data and set up their own instruments and things like that. And thousands of labs have just built these on their own. And this technology today is being used to make cell atlases of the, for example, of the human brain, but also of all the tissues in the human body. And people have kind of cleverly combined now droplets and beads with other kinds of things to do lineage tracing and pooled CRISPR screens and things like that. And although we maintain this sort of free open source version on our website, there have also been commercial adaptations that have further facilitated adoption of this technology by thousands of labs. So I'm just going to tell you about one story about something we've recently learned from applying this technology to a specific problem. And this is work led by Fenna Kreenan, a postdoc in the lab, who's really trying to answer the question about evolution and what is different about regions of the brain, like the cerebral cortex and the striatum. What's different about it from a human and a mouse and a primate? And what are the aspects of the human brain that you can model in a mouse? And what are the things that you would could never see in a mouse because it doesn't have the same cells? Or what aspects of human biology do different animals model? And actually, there's kind of great precedent for the value of thinking about these kinds of questions, even in the retina. So, for example, you may not know, but actually the photosensitive cells that detect light in our retina and the rods and cones actually evolved from deep brain photoreceptors in the pineal gland and the hypothalamus that are shared in lots of other species that don't actually have eyes. And so they were sort of repurposed to do this new job of vision, but they had been kind of sitting around doing something else for a long time before that. So, in this work, we actually focused on interneurons. So interneurons are extremely diverse. Even just looking at pictures of them, they differ. There's many different types with very different morphologies and physiologies, and yet they seem to be conserved. So that is to say, in many different species, including not just humans and mice, but turtles, people have looked and seen kind of like all the main kinds, and also seen them in kind of this repertoire being used across many different brain regions. So in this work, what we did is this is a comparative study of interneurons in humans, macaques, marmosets, and mice. And in each of these species, we used DropSeq to analyze tens of thousands of individual interneurons, enough that you could identify both rare and common, classify them into groups, and identify and learn from both rare and common types of interneurons. And this emerged in part from a larger project together with our colleague Guoping Feng at MIT, who has been working to develop marmosets as a model organism for translational neuroscience, and an NIH effort to make NIH-funded effort to make a molecular and cellular atlas of the marmoset brain and in fact of the brains of many species. So how might brains evolve if we just think about how does a brain change as you go from a mouse from an ancestor to a mouse or a primate or a human? So you can imagine any or all of the following things at work, and I'll give you kind of quick examples of each of these that we've found. So one thing that might change is just a specific type of neuron, or might be conserved across species, but just one species just decides to make more of it. So different abundances of types that are fundamentally conserved. You might have the same type, but it might acquire new jobs or functions in a different species, which might be manifest through a different molecular composition or set of RNAs that it's expressing. And finally, and most provocatively, a species might just find a way to invent a whole new type of neuron. And so adult cortical interneurons are distinguished by their embryonic origin. So interneurons are immigrants, and it's really good that we don't build a wall because otherwise the cortex would have no sources of GABA or inhibition or synchronization and pace setting of circuits. Interneurons are born in these places called the ganglionic eminences, and there are different ganglionic eminences that produce different types of interneurons. And at first, I'm just going to focus on at the highest level, which is just the median ganglionic eminence, which is the source of several kinds of interneurons, and these other ganglionic eminences, which are the sources of other ones. And we can readily tell where an interneuron came from the portfolio of RNAs that it's expressing, because there are really strong markers for these different things. So one of the differences across species that just jumps right out. So in a mouse, you can look at its frontal cortex and its visual cortex, and you see the same kind of quantitative ratio of interneurons that are derived from the MGE and interneurons that are derived from other eminences. But it's very different in primates. In primate cortex, it depends a lot on where in cerebral cortex you look. And in particular, in regions of the cerebral cortex that have more associative functions, there's increased representation of interneurons that immigrated from eminences other than the MGE. Whereas in V1 and regions that have more primary sensory functions, there's a more mouse-like ratio of MGE and non-MGE immigrants. So primates appear in their association cortex to have added many more non-MGE interneurons, and this is consistent with the sort of expansion of the upper layers in these regions of the neocortex that subserve association functions as opposed to primary sensory functions. So the second potential thing that could happen is that even for a conserved subtype, it might, in some species, express different molecules and have acquired new roles. And we find many, many examples of this. And in particular, here now, we'll go one level more detailed in terms of this interneuron classification and talk about sort of 4 large groups, 2 of which come from the MGE and 2 of which come from outside the MGE, from the other eminences. But these 4 groups are also readily recognized by specific markers, and we see all 4 groups represented in all 4 species that we looked at. But one thing that became quite clear is that many specific genes have acquired different gene expression patterns during evolution. So for example, neuropeptide Y in humans and other primates is highly specific to somatostatin interneurons relative to these other groups of interneurons. But in mice is substantially less specific and is expressed in an appreciable level in multiple kinds of interneurons. Netrin G1 and axon guidance Q also we find in the primates highly specific to ID2 positive interneurons. In mice, much more broadly expressed among the interneurons. Osteochrin, which is this this work that the Greenberg lab has done some very interesting work on, actually not expressed really in interneurons at all in mice, but in primates starts to be expressed in interneurons, and in fact in all the different primates in very similar patterns in terms of which kind of interneurons are using it, suggesting that at least among the primates that evolution has cared to maintain that pattern. And this is another example of just more quantitative evolution, one of the subunits of the L-type calcium channel. So even within conserved cell types, genetic programs evolve on evolutionary timescales. So finally, and most provocatively, I want to present an example of the just invention of a new cell type. And in fact, the example that I'm going to give is although we have found an example in the cerebral cortex, the example that I want to give actually comes from the basal ganglia, this structure that Yale talked about somewhat in her talk also. In part, and I want to emphasize this example because in some ways it's the most surprising. The basal ganglia is not where people go looking for novelty. So it's often described as the kind of deep reptilian part of the brain. It's not like the kind of the cerebral cortex that kind of appeared and then got really big along the primate lineage. Mice have it. Actually, fish have it. And in fact, the main classes of interneurons that have been, or of striatal neurons that have been identified in mice have also been seen in for example, lampreys. So again, it's not where you normally would go looking for novelty, but much to our surprise, we found it there. So here in the analysis that I'm going to show you, we've taken tens of thousands of mouse RNA expression profiles from tens of thousands of mouse striatal interneurons and tens of thousands of marmoset striatal interneurons, and then just aligned them computationally to each other. And you can see that most of the kinds of interneurons are represented in both species, but there's this very conspicuous set of interneurons that are represented only in the marmoset and not in the mouse. And these express markers that, again, are not normally seen in interneurons in the striatum. And I want you to notice it's not a trivial fraction of the interneurons in the marmoset striatum. It's about a third of the interneurons in the marmoset striatum. These are definitely seem to be their own type. They may be sort of most similar to parvalbumin and TH positive interneurons, but they're still quite different. They express their own complement and their own combination of transcription factors, distinct from those that are expressed by other striatal interneurons, occupy really specific places in the striatum, and finally, we find that they're conserved in humans. So when we do the same analysis now comparing marmoset interneurons to human rather than mouse interneurons, these guys reappear, but they have a very clear analog in the human striatum. And again, in humans, as in marmosets, they represent roughly a third of all interneurons. So they were always there, kind of hiding in plain sight, but you needed to know molecules and markers to be able to know, to be able to find them and see them there. And that's where we hope that these kind of unbiased systematic approaches may be able to kind of stretch our appreciation of just what's there in different regions of the brain, and also of what's not there in different model organisms. So this work was led by Fenna Kreenan, a wonderful postdoctoral fellow in the lab, and was a close collaboration with many other labs, including Guo Ping, who I mentioned at MIT, and Gord Fischel, our local resident interneuron expert. So I want to thank you for listening to this, and I'll be excited to hear people's questions and comments.
Well, I just want to say thank you so much, Steve. That was a really amazing tour de force and wonderful talk. I think there's time for a couple of quick questions, and then we have a coffee break. But yes, please.
Hi, thank you very much for this wonderful talk. It was really inspiring. I was actually curious to hear your thoughts about how well this single-cell RNA sequencing, or the drop-seq, for example, can represent well the different states. You mentioned that you can select out cell types and cell states, and I was wondering, for example, in the case of glial cells, how well that is conserved during this process.
Yes, so the question relates to what extent through these kinds of data can one see not just cell types, but other aspects of a cell's life. For example, the kind of physiological state that it might be in at any moment in time. And so I'll try to answer that through a couple of examples. So where there are transcriptional states that are known about, that you can say, do we see something that's known? The answer is just definitely yes. So for example, neurons, after they're electrically active, have been shown to express these immediate early genes. There's transcriptional cascade involving immediate early genes and other genes downstream of that. And in every one of these experiments, among the neurons, we can see a subset, usually just a small subset of the neurons of any type, often just 1% or so, but that have turned on those kinds of genes. So there is that specific transcriptional state, definitely is manifest in this kind of data. I think a really interesting question, though, is what we actually know about transcriptional states in neurons is actually quite limited, right? We know about some stress responses and immediate early responses. And so I think it's an interesting question whether it might be maybe possible to use data like this to kind of learn more about what's the sort of complete repertoire of gene expression programs and transcriptional states that neurons can execute. Then the other thing is in the area of microglia. We actually just had a paper came out in Immunity a couple of months ago together with Beth Stevens' lab, finding really exuberant diversity of transcriptional states among individual microglia sampled from the brain, especially during development. Less so during adulthood, but really exuberant microglial diversity during development. And often corresponding to, for example, some are microglia that seem to be in a high phagocytosis state, that are sort of particularly enriched along axon tracts, and maybe we're kind of like nibbling back, aberrant axonal processes and things like that.
I think the person in the front.
Thank you for the amazing talk. My question is about whole genome sequencing data. You used it for C4. I would like to ask you your opinion about preliminary results regarding the use of whole genome sequencing data to recover the missing heritability. It worked well in a recent paper from Fisher Group for BMI and height. Do you think it will happen the same in psychiatric disorders? Like we will be able to recover the missing heritability for these traits?
So I think the question relates to the kind of the missing heritability, which is I will sort of paraphrase as, genetic studies have found many influences on risk of psychiatric disorders, but still if you try to kind of make a predictor for them, they're not that predictive. So they help us identify molecular influences, which may be valuable for just pointing biology in productive directions. But it's sort of like many small influences, and so why can't we just sort of predict it, right? And so that's the kind of missing heritability question. It's like if these illnesses are heritable, why is there all this heritability that you just can't explain yet? And I think part of it is just that those genetic influences are only partially found. They don't suddenly become visible and obvious because you're whole genome sequencing instead of doing SNP arrays. So I think it's just, what does reveal them, whether you're doing sequencing or arrays, is just larger sample size. And it's just because these are complex disorders with many different influences, it's a big data problem. And as Amazon and Google and as everyone has figured out, it takes vast data sets to roll problems like that. So the key thing is the expansion in size of the genetic studies. It's not whether a study is like using super fancy whole genome sequencing or SNP arrays. One more question, Carrie.
Hi, Steve. Great work. Thank you so much for all your tremendous contributions. Sort of on that same point, if you could help us think about, particularly with the C4 example, still the relatively small effect size of 0.05 or 0.1 in schizophrenia, and yet the very robust apparent causal effect size in the model systems. How do we deal with these disconnects among small effect sizes in GWAS, but then targets that particularly are very robust?
Yeah, that's a really good question. And again, just to paraphrase, so in humans, C4 is just one of many genetic risk factors. It's just a quantitative contribution to risk. Why is it that in a mouse you can do an experiment and sort of and see such a robust phenotype? And I think this is a really important thing to understand. So it's why human genetics is just so different from mouse genetics. So mouse genetics, you have the typical experiment, all the animals have the same genome. You change just one thing, right? So you just make a mutation on an isogenic background, and the lab mice are completely inbred, so nothing else is different mouse to mouse. And then it's a controlled experiment, so they spend their whole lives, in a cage, getting fed at the same time, and so you've made everything else the same and isolated the effect of just one thing. And in human genetics, obviously, it's never like that, because every one of us, any two people differ at tens of millions of sites in their genome, and they have completely different life stories and environments and everything like that. And so it's very easy in a mouse for something to explain 100% of the variance that you see in the assay. It is not reasonable to expect it to do that in humans unless it's such a sledgehammer effect that nothing in the world can overcome or modify it. And so that's just why things are in general kind of so different in human genetics from most kinds of model organism genetics.
Well, I think on that note, I think just a big thank you for a wonderful talk. Very much appreciate your ideas. And just one thought, I loved your idea of the immigration without the wall. So immigration, schizophrenia interneurons. I'm just wondering, do you think that schizophrenia is an interneuron immigration disorder?
Well, we don't know enough to know, but I think that it's newly possible to do the kinds of experiments that ask that systematically. So I'll just say something, briefly about what we're doing in this area. Is and would like to do it at a much larger scale. So we're doing these single cell profiling experiments, but in postmortem brain tissue from schizophrenia patients and controls. And in each person now we can quantify in any region of the brain what's the abundance of each cell population. And so if all interneurons, or even if any one type of interneuron is consistently underrepresented in patients relative to controls, then I think that kind of thing will actually start to become clear, even if it's just a quantitative difference. And I think before you could have seen something like this if it was dramatic, and if you stained tissue with the right marker, and if you had the patience to go do that staining in hundreds of people and things like that. But that, this is sort of very hard to do. I think what's newly possible is to just sort of systematically ask that question about all interneuron types, and in fact about all, about microglia and glial types and everything like that, about all cell types, about all molecules, and then just do these kinds of things systematically in enough people that you can see it even if it's not the same cause in every individual person. And I think that's ultimately how we'll get to kind of the next level of insights about what, it must be in the tissue somewhere, right? There must be differences in the tissue, even though they're not visible to standard pathological analysis. And it's just a matter of I think these things will become clear, but it will take many people because it's not going to be the same in everybody.